Now, hello, I am Jacob Underwood, and I'm going to be doing the first problem, second problem, and third problem, and then my other group partners will be doing the others. We have Nathan and we have Joshua. So the first question is talking about a couple of different sequences that were given percentages for. So we just took the first one. We have A is 28%, C is 22%, G is 24%, and U is 26%. Now, we're trying to find out whether this is DNA or RNA, double-stranded or single-stranded, and circular or linear. So first of all, RNA, we know requires the U and doesn't have the T. So we can automatically assume that this is going to be RNA because of this. And then we know that double-stranded DNA will have equal parts of both the reciprocal. So in this RNA, A and U should be about equal, and G and A and U will be about equal, and C and G will be about equal. And we see that's the case. So we can assume that this is also double-stranded. Now it says that this is resistant to exonucleases, but sensitive to endonucleases. And this means if it's resistant to exonucleases, it's because it's in a circular form. So we can assume that, again, this is circular. If it's sensitive to both, then that would be linear. So the next answers that we would have for this, which is A to 34%, C to 37%, G to 14%, T to 0%, and U to 15%. That would be RNA because as U, single-stranded because they do not have the matching reciprocals, C to G and A to U, and it would be linear because it's sensitive to both. The next one would be DNA, double-stranded and linear, and the last one would be DNA, single-stranded and circular. The next question is filling in the blanks. We have DNA gyrase is composed of 4 protein subunits and uses ATP to compact bacterial chromosomes, while a nucleosome contains 146 to 147 base pairs of DNA and 8 histone proteins. The DNA double helix is 2 nanometers wide. Adjacent base pairs are separated by 0.34 or a 3400th in length nanometers. In the DNA double helix, topoisomerase introduces positive superlunar coiling into DNA and cuts 1 DNA strand. The origin of replication in E. coli is the ORIC. In the Hershey-Chase experiment, the bacteriophage 2 proteins were labeled with a radioactive isotope of sulfur 35, where the bacteriophage was labeled with a radioactive isotope of phosphorus 32. The sugar in uridine is ribose, and the sugar in deoxythymidine is deoxyribose. Now, for the answers to the third question, how long is the DNA portion of this translocation chromosome in meters? It's giving us the length of the amalgamation of the two, and we know chromosome 14 is 109 million, and 21 is 48. So we have to add those together for a total of 157 million, and then to find the length, we multiply that by 0.34 nanometers, and we get our length, but they want this in meters, so we multiply to the negative 9 to get meters, which is 0.05338 meters. How many nucleosomes are found within this translocation chromosome if we assume that the length of the linker is 50 base pairs? So we know 146 is approximately the length of one of those nucleosomes, and then we add that 50 to 196. So 157 million divided by 196 is 978,982. How many H2A molecules would you find in this translocation chromosome? So you take this number that you got right here, 798,000, and then you are going to be multiplying that by 2 because for each nucleosome you have 2 of those. And that will give you 151,597,964. And then finally, how many radial loop domains? This one was a little bit weird because radial loop domains can be between 25,000 and 200,000. So we took a grouping of how many in between it could be. So you divide 157 million by both of those, and you get the total of 785 to about 6,280 radial loops, somewhere in between there. Now Joshua will be going on and explaining 4.
Awesome. Thanks, Jacob. Okay, so you can see that. So we have a bacterium. Mycobacterium tuberculosis has a single circular chromosome that is 4,410,000 base pairs in length. If the chromosome was stretched out linearly, how long would it be? So you take the base pairs times by their length, and times it by times 10 to the negative third to get micrometers. So that's how we got that value there. Then we take that value for the next question, which asks for the diameter. So since we have circular DNA, we're going to take the circumference and divide by pi to get diameter. Right there. All right, so question C is this tuberculosis is organized into loop domains, with each loop domain with 50,000 base pairs. And if one of these was stretched out, how long would it be in micrometers? You take this number up here, that's how many base pairs, and divide it by so we can take this number and times it by the length of each, and times it again by times 10 to the negative third, and we get 17 micrometers. And how many histones are found? We can take this number of base pairs, divided by 196.5, and get that number because there's 146 to 147 base pairs of DNA per nucleosome, and so we kind of round it into 146.5, and then we add 50 because there are 50 base pairs in between each nucleosome. And then you times by 8, because that's how many histones are in each nucleosome, and then we get 179,542 histones. All right, so now I'm going to pass it on to Nate for
Part 5. I'm going to do this one a little more up close because I didn't want to draw every last structure of DNA that's written out here on the board. So the first question is, which of the molecules in part A through F are nucleosides? And so for that one, we just need a nitrogenous base, a 5-carbon sugar, but no phosphate group. And then, so the ones that have that are A, E, and F. And then the next one is which ones are the nucleotides. And so if it's not, well, on this list, it's either nucleotide or nucleoside. So those that weren't are B, C, and D. And then the next section is which ones contain ribose, which ones contain deoxyribose. So ribose has a hydroxyl group on the 2' carbon, while deoxyribose only has a hydrogen there. And so for the ribose, C, E, and F are ribose, and A, B, and D are deoxyribose. And then the last one is which one contain pyrimidines. And so pyrimidines has to deal with the nitrogenous base, which is that portion right there. And these are both purines because they have 2 rings, but this is a pyrimidine because it has 1. And so do the rest after that. And so the ones that have pyrimidines are C, D, E, and F. And that is week 7.